Alrighty. So, yesterday we finished building our DNA molecules, and then we started on this process of DNA replication. We talked about how, or not necessarily how, but why DNA replication needs to happen. Because if we don't replicate our DNA, then our cells cannot reproduce. So we can't have mitosis without getting a new strand of DNA. I'm going to briefly go over the stuff we talked about yesterday, just to make sure that it's all fresh in our heads. So we started out with what DNA replication is. It's just the process of getting two strands of DNA from one strand of DNA. And then we talked. Oh my goodness gracious! I guess that's what I get from sitting at my desk. Huh? Hopefully those still work. Whoopsies. All right. We reviewed the cell cycle from the very, very, very beginning of our cell unit. How we had our cell cycle. We had the M phase, G1, S, and G2, and the S phase is where we are existing within DNA replication, and that we can use what we know about DNA structure and our base pairing rules to figure out how DNA is replicated. We talked about our key players here: DNA polymerase, DNA helicase, DNA ligase, and DNA primase. Did I make y'all say polymerase? Okay. So I know they sound like big words. I will not ever judge your pronunciation if you choose to answer a question in class. They are big words and not very familiar words. But the more we interact with them, the more we'll get comfortable with them. So we had DNA polymerase, DNA helicase, DNA ligase, and DNA primase, all of which are proteins or enzymes that help carry out the process of DNA replication. Because without them, we can't replicate DNA. DNA helicase was the first enzyme we talked about, and it is the one that unzips our molecules down the middle. So we have our covalent bonds that hold the backbone together, because we need those strong covalent bonds holding our backbones together. But we needed weak bonds down the middle in order for helicase to snap it apart. What were those weak bonds called? Hydrogen bonds. Yes, I know it's up here. But hydrogen bonds down the middle hold our two sides of our ladder together. In order to duplicate our DNA, we have to break it apart. So DNA helicase does the breaking. If you ever get the enzymes mixed up, when you think helicase, think helix, because after DNA helicase does its job, our helix is no longer a helix. Right? So helicase messes with the shape of our DNA. It destroys the helix. But it can always come back together. We talked about priming, how DNA polymerase is kind of clueless until it knows where to start. So DNA primase adds a primer to the open faces of our DNA. So if we had our two strands of DNA, we ripped it down the middle. In order for replication to start, we need a primer, just like you would prime a wall before you painted it. I think this is roughly where we left off yesterday, so I'm going to go into a little bit more detail here, because this is the most important step of the DNA replication process. So if we have two strands, or if we have a strand of DNA that was split into two, right? If we had one like this, we had one way up here, like this, and they came together towards the end. DNA polymerase is what's going to add to those open-faced strands. So if we've got our little primer, our DNA polymerase is going to start wherever that primer is and move, adding free nucleotides. So nucleotides are just floating around in our cell to these open faces. So if right, our DNA polymerase is going to read those and add the appropriate, I used the wrong color, add the appropriate bases to it. So, if the original DNA is a T, what letters the DNA polymerase going to add to it? A, T, G, C, G, A. Beautiful. So DNA polymerase is going to add those together. The covalent bonds are going to form around the outside as they go, and now we have a whole strand of DNA, right? But the way DNA works, right? Our backbones are pointed in different directions. You remember that from our structure, where the little pentose sugars are pointed up on one strand and down on the other? What was that word called? Anti-parallel. Because DNA is anti-parallel, we have to build it in different directions. So DNA polymerase can only go in one direction. It can only move 3' to 5'. So because it can only go in one direction, one side of the DNA, so the 3' to 5' strand, right? It's just gonna grab onto that and go and not stop. But on the other strand, it's gonna have to start and stop and start and stop and start and stop in order to keep up with the DNA helicase that's going to do the unzipping. And I'll show you a picture in a minute. So we call this the leading strand and the lagging strand. So the leading strand is the one that can be replicated continuously, and the lagging strand is done in fragments. So let me do a little doodle for you. So if we have our strand of DNA helicase is going to come in and break what type of bonds? DNA helicase breaks hydrogen or covalent bonds. Just shout it. If it splits it down the middle, are those hydrogen or covalent bonds? Yeah, 50-50 shot. What bonds hold the two nucleotides together? We just said this. Hydrogen. Yes. Okay, so DNA helicase is going to snap the hydrogen bonds moving in this direction. The point at which DNA, or the point at which helicase is splitting, so this like little box right here, is actually called the replication fork because there's a fork in the road. So DNA polymerase can only work 3' to 5'. So if we have 3' to 5', and then 5' to 3', those are our two strands. So DNA polymerase can latch on here and go this direction. And that's fine. But on this top strand, it's going to have to go the other way. So if it starts right here, it has to go this way. And as it's going one way, DNA helicase is going the opposite way. So as DNA helicase keeps unzipping, DNA polymerase is going to have to start and catch up and just keep going back and forth and back and forth. So we have a bunch of different fragments. So if this is a fragment of DNA, this is a fragment of DNA, there's going to be a gap right here when this polymerase is gone. So it's done in chunks, essentially. And these chunks are called Okazaki fragments after the person who discovered them. Anybody have any questions about this? I know it's complex and it's kind of hard to visualize. Honestly, I think one of the biggest skills in biology is learning how to take something that is 2D and imagine it in a 3D space. Or taking something that is just described and creating an image in your head. So we're going to watch some stuff. I've got a couple gifts for you. How we doing? I hate this too. I get this. Come on. I won't be offended. Okay. So, Okazaki fragments. Here is another image of what's going on. So, but this is going the opposite direction of this one. So if DNA helicase is here, we're flipping it this way. We're unzipping it. DNA polymerase will be like right here, and it'll move. And as it moves, we're building our other half, right? But DNA polymerase is going to move this way on the lagging strand. So we've got a DNA polymerase starting here and going, starting here and going, starting here and going, and we have created these Okazaki fragments. So the leading strand is going the same way as helicase. It's just going to go and go and go and go and go. And the lagging strand is going to have to stop and catch up and stop and catch up and stop and catch up. Cool? All right. So that's a leading strand versus lagging strand. But we're not done, right? We've got a bunch of fragments attached to one half. How on earth is this going to wind up to different segments of DNA? That's where DNA ligase comes in. DNA ligase goes through and connects all of our Okazaki fragments, and that's really tiny font, so I will fix that. That's gracious. Okay, yeah. DNA ligase goes through that lagging strand and connects all of our fragments. They're like the glue that fixes everything, makes it all nice and cohesive. At this point, also, if there are any mismatches, like if a T gets matched with a G, right, that doesn't belong there. Sometimes DNA makes mistakes, so they have a proofreading. So DNA polymerase will also go through, reread it, and if there is a mismatch anywhere, they'll take it out and fix it. So there's a proofreading thing, just like we make mistakes as we are writing anything. So does our DNA. It's not a perfect system. Nothing in biology is perfect. A lot of it's overcomplicated and messy. So the DNA replication system also has a way to proofread kind of ingrained in the system. So DNA ligase will go through and connect all of our Okazaki fragments on the lagging strand. Bless you. Fix this. All right. So that's our proofreading step. And finally, we are left with two identical strands of DNA. But there's another vocab word in here, and that is semi-conservative. The process of DNA replication is considered semi-conservative because one strand of our DNA is conserved and one strand is brand new. So whenever we had our original strand of DNA and it was split in half, right? Once we build our new strand of DNA, it's going to be half old and half new. So every strand of DNA is half new and half of what the strand before it was. So if you had a cell, right, and that cell replicated, each one of its daughter cells will have half of its DNA molecule be the exact same one as the original cell, and half of it will be new. So we've got our original DNA strand, half of it's going to be newly synthesized DNA, and half of it is going to be that original template. So it's not like your friend did all the homework and you just copied it down word for word. It's like if you got a homework assignment, you said, hey, you do odds, I'll do evens, you come together, right? Half of it's stuff you did, half of it's stuff that your friend did, right? It's not your friend doing it all and you copying word for word. If that analogy is relatable to you at all. Any questions about this? I'll give you a second to get it down. Yeah, okay. So, oh, sorry, did anybody, is anyone still writing? Okay, yell at me if I move forward too quickly. So here is an image of the whole process going down, right? So DNA, right, wraps around histone proteins, and we get a chromosome, right? So if we unravel a chromosome, we've got our DNA. If this is our original template, DNA helicase, right here, is going to come through, snap all those hydrogen bonds, and break that helix up. Helicase breaks down the helix. Sorry, I think I'm going to sneeze. Nope, okay. Then we have DNA polymerase here and here, going through and adding our free nucleotides to our template. So our free nucleotides are chilling over here. They are just bits of old DNA that we've broken down and we're going to repurpose into new DNA. We have our leading strand that is going to go the same direction as our helicase, so it doesn't have to stop. And we have our lagging strand going the opposite direction, which will eventually lead to a bunch of fragments. Anybody have any questions? Is there a specific portion of this that you just don't get? Is there something that I can re-explain to you? Questions, comments, concerns? All right. So, when it comes to really, really conceptual stuff like this, I don't want to be your only source. So we're going to watch a video really quickly just so somebody else can explain it to you as well. Make sure it really gets in there. And then I've got a couple other small things to show you. And that'll be the day. Yes. Not this. Not who. As of right now, no. Let me check my little I don't technically do quizzes. We'll just do like activities based on it. Right now, if we do, it'll be next Friday. So it'll be what we learned this week and what we learned next week put together. So there's that. I'm going to pause the video recording for those of you at home. Yeah. Oh, wait, I have one more slide. Sorry, I forgot I added this. Prokaryotes and eukaryotes are different. Remember, prokaryotes are our cells that don't have a nucleus, so their DNA is a circle, whereas our DNA is just one long string. That's why we need a nucleus to protect it. So the way we replicate DNA looks different in prokaryotic cells and eukaryotic cells. In prokaryotic cells, we have it's just one circle, so we're only going to have one point where we start. So our helicase is going to pick a spot. It's going to go in different directions. It's going to start in one place. But on a string, it's not going to do that. But to save time, it's going to start in multiple places along the string. So if we had the pictures that we've drawn so far, if we zoomed out to see the whole DNA strand, we'd see a bunch of those images all lined up to one another. They're called replication forks, is where we are actually doing the replicating. And the origin of replication is wherever it started. So if we had an origin of replication here, here, and here, it would just keep going until they all met, which is why we need DNA ligase in the first place. Anybody have any questions here? Just real quick, prokaryotic versus eukaryotic. Okay. All right, I'm going to pause this. I'll be back. Resume this. Perfect. Okay. So I've got one last image for you before we get started. If we were to take a tiny, not tiny, but a very, very powerful microscope, this is what we think we would see. Actually, we'll leave this off. This right here is DNA replication happening in our cells. It looks really, really wonky, but if you can see down here at the bottom what's happening, we got a lot of proteins involved, right? So this is our original DNA strand right here, and it's being split into two. One is going this way, and one is going this way. As you can see, we'll just keep on waiting. This one right here is just going and going and going and going and going. It's our leading strand. We've got a DNA polymerase right here that's adding all of our nucleotides. But this strand right here has a lot more stuff going on. That is our DNA. That is our lagging strand. So this little green guy that flies in here is going to be our primers or our primase. So if that helps kind of put it into a 3D model for you, we got that. So whenever we think about DNA replication, step 1 is to break it apart. Step 2 is to get our primers intact. Step 3 is to actually do the replicating, have DNA polymerase fly through there and add our free nucleotides. And then we have our proofreading and fragment put together that DNA ligase has. Anybody have any questions? Okay. I'm gonna keep checking up on you because if we get lost here, the next thing we learn might be a little harder. So if you are at any point, you have any questions, if you're just lost in general, please let me know. I'm more than happy to help out. There is an exit ticket on Canvas. If you could fill that out for me, please. You have a little extra time, so please take your time with it. If you haven't done any of the other exit tickets that we have done so far this year, go ahead and do that as well. I want to make sure that y'all have what you need done. It is only week 1, so if we start knocking these out now, then we won't fall behind by the end of the 9 weeks.